Hello guys and welcome back to Excelsior Academy. So in the previous lecture, we were discussing about the actin filaments, right? So in continuation of that, let's talk about the contractile assemblies of actin and myosin in non-muscles. So अब तक हमने muscle cell में देखा, skeletal muscle में देखा like how the actin filaments are helping and how the contraction is happening. Now same thing in the non-muscle cells, like normal cells में कैसे होता है, ठीक है? फॉर एग्जाम्पल ये नॉन मसल सेल्स कैसे हो गए, जो आपके लिवर सेल हो गए, किडनी सेल हो गए या WBCs हो गए. Okay, so like how movement or the contraction is happening in these particular type of cells. So the actin myosin filaments in non-muscle cells are not organized into sarcomere, okay, but they do have the stress fibers and the adhesion belts. So remember, we have already talked about it, like what are the stress fibers and the adhesion belt. So whenever a cell is there, right, it is making certain focal adhesion points, right? So it is making focal adhesion points. So these are the adhesion belts. Now stress fibers, जो आपके actin filaments ऐसे present रहेंगे. Now the contraction of these stress fibers produces tension across the cell, allowing the cell to pull on a substratum. यानी कि क्या? It is helping in making sure to form a force onto this adhesion point so that it can pull. Okay, because it is attached to this. So एक तरह से इसको पकड़ रहा है और उसके हेल्प से वो आगे को बढ़ता है, ठीक है? So the contraction of adhesion belts alters the shape of the epithelial cell sheets, a process which is particularly important during the embryonic development when the sheets of epithelial cells fall into structures such as tubes. Okay, now look at over here. So this is how in the case of the the actin myosin, it is sliding, right? So there we saw the difference, like how the myosin and the actin are present, the sarcomere is present, and then the actin filaments are pushed together so that the contraction can take place. Here you can see the arrangement of the myosin. So though two globular heads are present over here and two are present over here. So then again they are having the myosin binding sites are present. And then when they are moving, what is happening? This actin is moving in this direction, right? And this actin is moving in this direction. So depending on the direction, like where the cell wants to go, okay, the actin filaments are going to contract with the help of the myosin. So let's talk about another example, that is cytokinesis. Now cytokinesis is also because of the actin filaments, okay? So cytokinesis means it is the division of a cell into two following the mitosis. So towards the end of the mitosis in yeast and the animal cells, a contractile ring consisting of the actin filaments and myosin 2 is assembled underneath the plasma membrane. Okay, so the contraction pulls the plasma membrane and finally what happens? It pinches off and it becomes in two. So this is how you can see. So we have got this contractile ring. Now with the help of the myosin, okay, with the help of the myosin, it is getting contracted slowly, and then finally pinches off. And what we get is at the end, two daughter cells. Same way the fluorescent image is shown over here. So the blue color one is the DAPI, which always stains the nucleus. The green color is your actin. Can you see it over here? This is the contractile ring. So along with the myosin and all, you can see there the contractile ring is slowly is getting shortening. Okay, and finally it pinches off. Now when I am saying the contractile ring is getting shortened, what exactly is happening? Yes, anyone? Akshay, would you like to try?
Are the filaments like again depolymerized so that it reduces?
Yes, little correct. See, what is happening? See, पहले आपने contractile ring इतना बड़ा बनाया, ठीक है, of your actin. Sorry. Okay. Now what is happening? The myosin filaments are making sure that this actin filament is getting contracted. Okay. So when it is becoming this much, so rest of the extra actin jo tha, okay, Now that is getting depolarized. It is not required. So slowly, it is becoming small, and finally pinches off. Okay, so yeah, it's correct. So a few of the active filaments starts depolymerizing. Okay. Now, in non-muscle cells and also in the smooth muscles, the contraction is regulated by the phosphorylation of one of the myosin 2 light chains, which is called the regulatory light chain. So if you see the structure of the myosin, we saw over here, the two heads are there, right? And then two light chains are there. Okay. One second, guys. But yeah. Okay, so these light chains are getting phosphorylated. Now, getting phosphorylated means what? It is getting regulated. Okay. Yeah. So phosphorylation of the regulatory light chain in these cells have at least two functions. First, it promotes the assembly of myosin into filaments, and it increases the myosin catalytic activity, enabling contraction to proceed. So as such, myosin, when it goes and binds to the myosin binding site on the active filament, it is not active, okay? We need phosphorylation of myosin molecules also. Then only it gets activated. Let's go a little slowly over here because this point is very important. Okay. So now what we have seen, so myosin is there, it is getting phosphorylated, okay, onto the light chain. Now, the enzyme which catalyzes the phosphorylation called, of course, it is going to be a kinase. कोई भी protein, कोई भी enzyme, when it is adding a phosphate group, it is going to be a kinase. Okay. So now this kinase is known as myosin light chain kinase. Simple. Which is itself is regulated by the association with calcium binding protein known as calmodulin. Okay, this is also important. Now, when there is an increase in the cytosolic calcium, it promotes the binding of calmodulin to the kinase, which results in the phosphorylation of the myosin light chain, regulatory light chain. Did you get it? So, जैसे ही non-muscle cell में there is an increase in the calcium 2 plus level. Okay, this results in the activation of the calmodulin. जितना ज्यादा कैल्शियम होगा it goes and binds to the calcium binding protein known as calmodulin. Now this is going to activate your kinase. कौन सा kinase? Myosin light chain kinase, eventually resulting in the phosphorylation of the myosin regulatory light chain. Did you get it? Now, increase in the cytosolic calcium are thus responsible for activating myosin in the smooth muscles and the non-muscle cells as well as in the striated muscles. So this is the path by which the myosin 2 is getting activated in the case of the non-muscle and the smooth muscle. Yes, is it clear, everyone? Krishita?
Yes, ma'am.
Now, this is how it is happening. See, most of the time in the exam, like what I have seen is the 4 marks जो इतने lengthy questions होते हैं ना, in that most of the time they don't write the full name or full form of the enzymes. Okay, like for example, ये क्या है आपका myosin light chain kinase है. Sometimes they won't write it, and if it is happening, like they are not writing the name of the enzyme fully, please understand that उस क्वेश्चन में उस एंजाइम का कोई रोल ही नहीं है. Did you get it? वो सिर्फ आपको कंफ्यूज करने के लिए इतना बड़ा सा लिखा हुआ है कि this is happening, this protein is doing this particular protein is doing that particular function, and the end में मगर पूछा कुछ और है. ठीक है? So there are also certain questions like where they mention a lot about the protein, but they are not asking about that particular protein. Then the question is something else only. We'll do all such questions also. Okay. Now look it over here. So first thing first. So calcium. First, the calcium is coming. There is an increase in the calcium level in the cell. It goes and binds to your calcium binding protein. Now, when you see that this calcium binding protein, calmodulin, the shape is different, right? The moment calcium binds, there is a conformational change. Now, this conformational change makes it your MLCK active by binding to it. Now when you see the structure, okay, this conformational change which is brought by the calcium binding, now we see that it is by, you know, getting change, because that's how it is going to bind and activate MLCK. Agar ye aisa hi hota, then the interactions would have been difficult, right? So nature understands like when and what to be activated and how it should be activated, right? So when this calmodulin is getting conformationally change upon the binding of the calcium, it goes and activates your MLCK. Now, when MLCK is binding, you see that this gets open up. Ek tarah se if you see, you know, it's like a finger, it just came out. So this portion is present over here. Now, when this is activated, so this is now your calmodulin MLCK complex. This is going to add phosphate group to your regulatory light chain of your myosin. Can you see the regulatory myosin, regulatory light chain? ये जो अलग से कलर किया हुआ है, yeah, so they are getting phosphorylated. So from inactive myosin 2, it becomes active myosin 2. Now, among all the different types of the myosin, there are several types of the myosin. Okay, out of that, myosin 2 is playing very important role. It's like crucial in wherever the contraction we discuss about. Now, do you remember where we have talked about myosin 1? We have discussed about the myosin 1 previously. Any idea? Do you remember कहाँ पे हमने डिस्कस किया होगा? श्रुति? I don't know whether you were there in that lectures or not. Acha, Tanvi, would you like to try? Do you remember we were talking about the microvilli?
Yes, ma'am.
So in the microvilli, I have mentioned that there are the again actin filaments are there. Say actin filaments were present, and then each were bound with the help of fibrin and all, right? So here the myosin were present and again interacting with the actin, essay, for the microvilli movement. So here you have got your myosin 1. Okay. Anyways, I have to share all those videos also. You can go through then. Yeah. So coming to the unconventional myosins. So when I say unconventional myosin, we are going to discuss about the other types of the myosins. So they're not involved in the contractions, but they're important in the variety of cell movements, including the transport of membrane vesicles, organelles, and other cargo along the actin filaments. So if you remember in the video once I showed you, so we have got these microtubules onto which the cargo is being taken up by the kinesin and the dynein proteins, right? Essay. In the same way, we have got myosin proteins also carrying the membrane vesicles and organelles, and they do walk on the actin filaments. ये क्या था? ये आपका microtubules में था. Now, if you remember the videos, we have shown that actin filaments भी आपके ऐसे network बनाते हैं across the microtubules, right? And they do make a connections with the like suppose this is your actin filament, they make small connections with the microtubules also. Again, same analogy, like all your कच्चा रास्ता जो रहता है eventually कहीं ना कहीं तो वो national highway को join करता ही है, right? In the same way, actin filaments are also widespread present. All these organelles move, and finally they reaches to the microtubules for the final destination. Okay. So myosin 1 proteins, they contain globular head that act as a molecular motor, and the tail binds to the other structures such as membrane vesicles or the organelles. Okay. So the movement of the myosin 1 along an actin filament can then transport its attached cargo. Let's see the figure. Yeah. Can you see small myosin 1? So the head is present to make interactions with the actin filaments. Okay. And the tail are attached with the vesicle or any other organelle. So you can see that, you know, new myosin is coming, it is making an attachment, and with each attachment, ATP hydrolysis is required. If you remember when we were discussing about the myosin 2, actin and myosin 2 in the sliding filament theory, with each movement of the myosin head, globular head, ATP was required so that it moves and binds to the another myosin binding region in the actin filament, right? So for each interactions, ATP hydrolysis was required. In the same way, here also, if you see here, again, myosin head is present. ATP hydrolysis happens, it leaves and it joins to the another version, right? And the tail is again attached to your membrane vesicle, and slowly it moves in this direction. Now, please remember, whenever this movement is happening, like अगर आपसे question पूछे कि myosin कौन से direction में जाता है, then the answer is plus end. ठीक है? Now, same way, kinesin जो microtubules motor proteins है, kinesin also walks towards the plus end, whereas dynein walks towards the minus end. ठीक है? क्योंकि कुछ चीजें जो अगर आपके बाहर जा रही है, like for example in a cell, if you have got those centrosomes, so microtubules आपके ऐसे बन रहे हैं ना, ये plus end हो गया. If something is going out towards the periphery, then there are chances that the vesicles which are formed newly, they should come towards the sorry, towards the center, right? So for that, we have got the dynein, which moves along the microtubules in the minus direction. Okay? Okay, we'll talk about microtubules also. Is this is clear, like how myosin 1 is helping in the movement of the vesicles?
Yes.
Yeah. Now, so the function of the myosin 1 is to help linking actin bundles to the plasma membrane of the intestinal microvilli. So this just now I explained like how in the microvilli myosin 1 is present. Okay, so these structures, the motor activity of the myosin 1 moves the plasma membrane along the actin bundles towards the tip of the microvilli. Now, additionally, it helps in the transport of the vesicles and the organelles along the actin filaments. There are other unconventional myosin which looks similar to the myosin 2. For example, myosin 5 is a which function as a two-headed dimer to transport vesicles and other cargo along the actin filaments. So this is when you know the example of myosin 5. This looks little similar to your microtubule motor proteins. Again, head domain is present. This actually moves like how we walk, right? Left and right, left and right. In the same way, even these motor proteins walk. Then they are coiled, they have got the light chain, and the tail portion is interacting with the intermediate filament. Now, why intermediate filament? Because these filaments are required at the plasma membrane periphery. Okay, so they are taking this intermediate filaments towards the end. Okay. So all of the myosins move towards the plus end of active filaments. Exception is myosin 6, which moves towards the minus end. ठीक है, ये भी आपको याद रखना है. Now these are the different types of the myosin. You can see like how the head and the tail is varying, right? So one हमने देखा, 2 is like, you know, there are 2 are there and they're coiled together. Then 3 single is a, it just has the extension at the globular head. Myosin 5, it is similar to your myosin 2. It's just that the difference comes in the light chain, light regulatory chain. Similarly with the myosin 6, again the difference is coming in the light chain. Then 7, 8, 9, 11, many are there. Okay. So this is just one example, like myosin 2 is involved in the actin filament where the contraction and cross-linking is happening. Okay, myosin 5, which is helping in the organelle tethering and the transport, and sixth one is helping in again the organelle anchoring and motility, actin filament organization. You don't have to talk about all these, but please remember like myosin 2 कहाँ पे उसका role है, myosin 1 का कहाँ पे है, and 5 where it is involved. Okay, is it clear if the question is being asked about the myosin 1, 2 and 5? Yes. Yeah. So why myosin 6 is also important? Just now as we discussed that it moves towards the minus end. ठीक है, बाकी सब आपका plus end है. Only myosin 6 moves towards the minus end. Okay, so with this actin filament is completed. Any doubts in this section? No? Okay.